Hello friends. So in this video we are going to learn about genetic code. So this is basically what we have studied. This is our continuation. This was our DNA sequence. From this DNA we got RNA. This we got in a consequence of complementary base pairing. We know these things, so I am not going to discuss again. This was DNA. This was RNA. So we got this in the basis of complementary base pairing. But how this RNA is producing me a stretch of protein or chain of amino acids? This is here comes into play the genetic code. So what happens? We have 20 standard amino acids. How many? 20 standard amino acids. So scientists initially thought that to know this 20 amino acids, these 20 amino acids are being coded by the nucleotides present. So if this is the DNA sequence, the complementary sequence will be like this: U, A, C, G. So scientists initially thought that one nucleotide sequence is representing one amino acid. So by taking this concept, we can represent only 4 amino acids. So this is not the exact representation because we are left with 16 amino acids. Then we thought that no, we take two nucleotides to represent one amino acid. If these are the two positions, in any position there can be have any nucleotide, and irrespective of this one, any nucleotide can come here. So we have 4 possible positions at this one, and we have another 4 possibilities at this site. So you know better about permutation combination. So we will have 16 possibilities. That means we can represent 16 amino acids by taking 2 nucleotides coding for 1 amino acid. So then also there are 4 amino acids are being left. So what that scientists finally did, they took 3 nucleotides are going to represent 1 amino acid, and when this was done, they saw that by doing this, we can represent 64 amino acids, which is much more than 20. This is enough. So this sequence of 3 nucleotides is my genetic code, finally going to represent 1 amino acid. So if you consider there's all nucleotides, that means U, A, C, G. Why am I using U? Because genetic code is the basis from RNA, not DNA. So RNA U, A, C, G. So the combination for now, we will get a table of 64 genetic codes. So I'm not going to represent those all, but one of the most important question in our board exam is this one, right? Salient features of genetic code. So this is very repeatedly asked in exams. So you please try to understand all these points. So these points are very much pretty easy. So don't worry about these things. Try to understand these 2 points very closely or very well. Genetic code is unambiguous. Unambiguous means that suppose you consider any nucleotide, suppose AUG. AUG codes for methionine. This is an amino acid. Methionine is coded by the genetic code AUG. So unambiguous means that AUG will always represent methionine. AUG cannot represent anything else. Side by side, we are going to deal with degenerate. Suppose G, 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 U, G, G, C, G, G, A. All these codons represent the amino acid glycine. Meaning is that glycine can be represented by more than one codon. This feature is called as degenerate. We can understand the unambiguous nature here itself. Meaning is that glycine can be represented by 4 codes. This is the nature of degenerate. And GGG will always represent glycine. This is the property of unambiguous. Or vice versa, or not vice versa, or simultaneously, GGC will always represent glycine. This is unambiguous nature. Reversely, glycine can be represented by all these 4 are the nature of degenerate. So don't mix this two things, very important. Universal means that AUG codes for methionine. This is for human being, bacteria, animal. You can take any living condition. AUG will always code for methionine in different organisms. This is the nature of universal codon. Commaless, that means if you have to represent any code, you do not have to put any full stop or comma or semicolon, anything in between. Here or even here, suppose this is G, G, G. I am representing G, G, U here. You should not have to represent comma here or even here, nowhere. You just have to write in continuous fashion. That is that they are commaless. Initiation codon by initiator codon, meaning is that any gene sequence will be starting with a codon called AUG that represents methionine. This is always a starting codon for the genes. And for stop codon, this will be more critically analyzed in the translation part. For this stop codon, there are 3 sequences: UAA, UAG, and UGA. So when the terminal sequence of a gene will come, at the end there will be these 3 sequences which we consider as stop codon, where our sequences will be ending and our gene sequences will be ending for the completion of the process. So these are the salient features of genetic code. You must remember the features. We will provide the link in our description box. Thank you so much.